think I scared everybody else off. A lot of talk. But I believe you guys should be through this already. Dr. B said you guys are starting chapter 9. And if not, I mean, this is a really good preface. I'll wait one more minute, just in case. I'll start sharing my screen, but by no means am I starting quite yet. Let's see. Okay, I'm gonna go ahead and continue. So when I was talking about the modes of transformation, or sorry, the modes of information transfer. I was talking about horizontal versus vertical, right? Vertical is strictly for reproduction, right? However, horizontal has one of 3 forms: transformation, transduction, and conjugation. Transformation, it comes best in the example of the experiment done here. So what happened was we injected mice with a heat-killed smooth pneumococci with capsule, a live virulent smooth pneumococci with capsule, and then a live non-virulent rough pneumococci with no capsule. So obviously the live virulent pneumococci smooth with a capsule kills the mouse. What we wound up seeing is that if we combined the dead version of that, right, the virulent kind was here, but if we did the heat-killed version, the mouse lived. So if this one caused disease and we killed it and injected it in a mouse and it didn't cause disease, then what happened here? We gave the rough pneumococci without a capsule, non-virulent, doesn't cause a disease. We mixed that with our heat-killed version of the one that does cause disease. What happened was a horizontal transfer of DNA. This horizontal transfer of DNA made live smooth, the virulent cells, right, the ones that killed this middle mouse. It wound up making those bacterial cells inside of the mouse with the live non-virulent rough pneumococci without a capsule. So these 2 bacterial forms were found because some took on the genetic information of the heat-killed and some did not. But these were both isolated from the mouse, which is very interesting. So how does this occur, right? Obviously, the cell has to be competent. The DNA fragments are now free-floating, they're raw, right? They are a pure form of DNA. The naked DNA enters the competent cell, and then all of a sudden the competent cell starts reproducing with that DNA now part of it. Okay? So when we did our transformation lab, right, we took naked DNA, we made a cell competent, and we mixed the 2 together so that we would have competent recipient cells with the DNA, and it would actively divide and recreate and produce the genetic engineering product that we wanted, right? It's hard for words today. But this, if you want an example, think about what you did in your transformation lab. That is transformation. It wasn't just a word to tell you that it changed. This is the type of horizontal gene transfer we're talking about. So that is the example there. For transduction, I know there's two trans and then conjugation, you'll know. But transduction is the transfer of DNA by a bacteriophage. So it will be discussed more, obviously, but what you really need to know is the lytic versus lysogenic cycles. You'll definitely need to know this for quiz number 4. Quiz number 4, this is a couple of questions on there. Oh, what? Not going to use this slide. I think that was just for me. But know the lytic versus lysogenic. What the functions are. You don't need to do step by step by step. Maybe drawing it out would help, but know the key differences between the two. Let's see. So what essentially happens is a bacteriophage is a virus that infects bacteria. Only bacteria. What will happen is the bacteriophage will carry DNA with it and inject it like a hypodermic needle, literally, inject it into the cell. And if we have this lytic cycle, it'll inject this DNA, it'll become part of its chromosome, and then it will eventually start to reproduce. And the whole point is that it's injecting its DNA to perform its own life cycle. So the bacteriophage will inject its own DNA to be reproduced within the cell, and then it bursts the cell, and then 5 more bacteriophages are born. That's how they reproduce. Okay. The lysogenic cycle, however, it doesn't involve the cell lysis. In fact, it will continue and continue and continue to reproduce the information that the bacteriophage gives it. Sometimes that can be good for the bacteria. It just depends. You'll need to know the difference between generalized and specialized, right? So an example of a specialized gene, let's look. Oh, there's no examples, but if I'm not mistaken, an example of a specific bacterial gene would be an antibiotic resistance. I mean, who doesn't want that? If you're trying to survive, you're like, hey, I'm now going to be immune to this thing that's like around me. Like, sign me up. I do not want to be killed by an antibiotic. So a specialized bacterial gene that is transferred via the phage, in the phage, is, it's special, right? But if you just so happen to stumble upon this bacteriophage and it has some DNA, and then all of a sudden you get like, I don't know, you get like a little bit bigger, right? The bacteria just gets barely bigger. It's like, oh, okay, this is kind of random, but okay. But just know that it's any versus specific, but you guys can tell by the names. Alrighty, so last but not least, conjugation. This is a truckload. Think of a semi of DNA. And this requires that sex pilus. So there are many, many videos that will show you this. This is the main picture. So you guys have to kind of think of which one has the DNA, which one doesn't. The one that has the DNA is gonna go and grab this other cell, and it's like, what's going on? The bacteria that has the DNA is gonna be like, come here, and is gonna draw this bacteria in. And then all of a sudden it's gonna whisper sweet little nothings in its ear, and it's gonna give it all the DNA that it wants. So then they fuse their cells, their cell walls, and all of a sudden they can give them all this information, usually in the form of a plasmid. And you know how I said that one cell is going to have the DNA and one cell's not? The cell that has the DNA is going to be considered an F positive cell. And the F positive cell means the fertility factor. So when the F positive cell gives the DNA to the F negative cell, right, the bacteria cell that does not have the DNA, when the F positive gives to the F negative, they both are F positive. And this is the sex pilus or the conjugation pilus. So that the bacteria that now got this new DNA can go do that to somebody else, whisper sweet little nothings, right? Go find another F negative cell. So there are different types of plasmids. I know this kind of gets confusing, but the different types of plasmids can tell the cell what to do in different ways. So, for example, the F pilus would be the fertility factor, right? The resistance plasmid would be something like antibiotic resistance, right? That'd be like MRSA. MRSA has a resistance plasmid. Virulence plasmids. Remember how we talked about that certain strain of E. coli that had this new toxin that made it even worse? That would be an example of a virulence plasmid that it got. The bacteriocinogen. So produce growth inhibiting bacteriocins affect strains of the same species. That one's not covered that much, I'll tell you that. But I guess just know the definition. But the three big ones are the first three. Let's see. You guys, I think you guys can figure out resistance. Resistance plasmids are called an R factor. Know that one plasmid can carry up to 10 resistance genes. So it can, let's say that you have a resistance gene just for penicillin, right? That would be just one antibiotic. But if you can have a plasmid that's resistant for penicillin, vancomycin, all these other antibiotics under the sun, that can be pretty detrimental, right? So it could be as many as 10 antibiotics it could be resistant to. It's, this is a very, the fact of the matter is that resistance to antibiotics is very high all the time. This is why I stress so much, if you are on antibiotics, finish them, or so help me God. Because all of a sudden you're breeding basically these resistant bacteria in your body. And who knows, you could give that to somebody else. And then they take either a similar or, hopefully they take a similar one, realize it doesn't work, and then take the full course of their next antibiotic. But what if you give somebody an antibiotic resistance bacterial and they take a different antibiotic than you did? They didn't take the whole course. All of a sudden it's resistant to that one too. You see how this can be very detrimental? The more we go, the more we spread, right? Nasty stuff. So you guys will learn shortly, I think next week, this beautiful thing called the Stanford Guide. You guys don't need to buy it. There's ones in the classroom and there's one online. What it shows you is it shows you bacteria and every single antibiotic under the sun. It'll show you the bacteria and all these antibiotics in a little cross grid, and it'll show you if it's effective or not. So if it's an effective antibiotic for that specific bacteria, it'll show you. And it's very important that we take wound cultures, that we take sputum cultures, we take everything we can to figure out what this bacteria is before we just pump you full of a really strong antibiotic or pump you full of an antibiotic that might not work. Now, if it's a dire situation, they will pump you full of antibiotics. If you're dying of sepsis, they will give you antibiotics. But if you just come in and you're like, man, I have a really bad cough, my throat's really sore all the time, like I can't deal with this, they'll swab your throat because they want to send that off to the lab and figure out what you have. Because also, I don't know if you guys have gotten to this part yet, but there's a certain chart of diseases that if you are detected for it, you have to report them. So let's say you go to the STD clinic, right? And you get a swab done and you figure out that you have syphilis. And that's one of the things that have to be reported to the government. Not that you specifically have it, but that there is a case in this location, because they want to track those things. They are considered highly virulent, and it's important statistically for us to figure out why and how these things are being spread, because, you know, syphilis can cause scarring on the fallopian tubes, and then all of a sudden, you know, people are infertile. And obviously this causes severe emotional, psychological damage. And that's, you know, something we obviously want to avoid in all of our patients, right? So enough of my tangent about antibiotic resistance, but let's see, okay, so there's plasmids, but there's also transposition, which is just, and it just kind of cozies its way into the gene, and then it just stays there, and then the bacteria copies it without even knowing. It's not just unique to resistance, to antibiotic resistance, so it could be different. It could be like the Indian corn, right? Let's see. This is a great slide. Does anybody have any questions? Okay, I'm gonna move on. I'm trying to get through this. I know it's a lot. We talked about generalized transduction, right? Versus specialized. I think she put these at the end because she doesn't really want you to know it that well. Yeah, these, no, don't know. If it's past, let me see, if it's past this question slide, you don't need to worry about it. I'm gonna stop that there. Nobody freak out. So that is that. You're okay. I'm going to reiterate it one more time. Okay. I love these resources. I use them religiously. All of these videos, I would redraw them out. I would watch them a million times. I would watch them until I figured it out. These are great, especially if you want to just review before the exam, review before the quiz. These are great videos, a great resource. I would encourage you to explore these. If you watch it and you're like, man, I know what this is, great. You check that off the list of what you need to know. Move on to the next one. These are all fairly short too. You can get through almost all of them within 30 minutes. I'm willing to bet. They're all, I think the longest one is probably 11 minutes, 12 minutes, maybe. I think that's it. But most of them are only like 3 or 4 minutes. And if you have like a minute or two to spare and you're like, man, I really need to study, watch a video. And then, you're like, oh, wow, I did a little bit of studying, I can do more. It's an easy milestone for you to, hold on to. Let's see. So we enjoyed the pasteurization lab. We enjoyed our take-home labs and our transformation lab. So you guys either did or will do our lab number 12 and 14 for the remainder of this week. And then next week, you guys will go back to lab number 12. But yeah, I think we ended right on time. I said I did the I was going to do dilution. That's right. So I drew out in class a nice little, I hope it's not flipped, but I drew out a nice little information review. I'll send a picture on Remind about what this is, but I'm trying to outline the difference between the dilution and the dilution factor, how to calculate it, all of that. I'm trying to give you guys examples and show you what forms the certain things will come in. So do you want me to walk you guys through that, or do you guys want to look at this and then tomorrow we go through this. What do you guys think? I bet you guys can't see it, but I'll post a picture, don't worry. So you guys burnt out today? Because I know I am. Are you guys willing to come tomorrow morning, 11:30 to noon? I will go over dilution if you guys want. Otherwise, I'll just post a picture, and then anybody who has questions can private message me, do whatever. And if I get enough people to have the same question, I'll definitely make a whole review session out of it. Yeah, no problem. Bye, Ling Ling.
So you wanted to meet tomorrow at 11?
11:30, yes. I do.
Oh.
All of my sessions are pretty much the same every week. I have the Monday one at 5, and that usually lasts an hour. Usually, sometimes we go over like 15 minutes, maybe. Then today's is always at 7. Wednesday is always at 7, and that's about an hour as well, sometimes over. And then Thursday is always 11:30 to noon. I kind of have to keep that schedule because I have to eat before class too, and I want to be respectful of that lunchtime as well, because I know people, they might get out of class at like 11, and if I'm already taking up that 11:30 to noon slot, I want them to be able to eat, be able to recuperate, maybe come to class with more questions if you wish to study even more between classes, whatever it might be. But I just wanted to give a lunchtime if there was not one already allocated. But yeah. And I'll always send out reminders too, about what's going on. Trying to think, anything else that I need to talk about. The only time that there will not be a PAL session is when there is a scheduled holiday. If you do not have class, then I do not have a PAL session. I want you guys to rest, and I want my own rest, right? If there's one day you can't think about micro, take it, right? Dr. B also wanted me to reiterate, make sure you choose your micro projects and or start them. Very important to get a head start. Case study number 2, get a good dent done if you can. And I believe that lab 8, 9, 10 are already done. They should be in. I think that's about it. Oh, I did want to mention that you guys are going to be starting your big unknown report. This unknown report, it's going to be, that's the 28-page mammoth that you guys have to look dead in the eyes. So what I would recommend you doing is, you're going to be getting information more and more. You won't know what you actually have as an organism until much later. But I would recommend that as you start doing more tests, make sure you take pictures of everything, and make sure that you maybe do a write-up and interpret it on said report. Because if you do that work now, you just have to format it later. It's going to save you a world of work. But yeah, so get a head start if you can on the on anything. Because if you pace yourself, you won't be sprinting at the end, and you'll feel good. But that's all I have for you guys today. I'll post that picture right now, and the lecture or the recordings momentarily. Okay. So you guys are free to go. Have a good night. Rest up, and enjoy the weekend if it's your weekend. If not, I'll see you in lab tomorrow. Okay, guys? No problem. Have a good night.